the projects that we are working on in the lab, one of the things that struck me when I was participating in the Genome Project, and I don't know how many people know this, I think a lot of people know this, the Baylor and the genome centers were competing with a private company which was going to sequence our genome and patent it. So one of the biggest challenges for us is to get it done before them. In science, we don't usually face this challenge. You usually take a problem, you wrap your brain around it, you think about it, and then you solve it on your own time. And for the first time, we scientists were faced with having to get something done in an unimaginably short period of time, a very challenging thing. And so what we had to do is we had to grow from a lab of 10 people at Baylor to 250 people on 3 floors on the top of the ALCAC building with 100 machines, with companies coming together with evolving technologies to match the ever-growing needs, and with conference calls with Washington every week to inform them of where we are, was a huge challenge. And we scientists, we don't have MBAs, we essentially ran a company because we had to deliver on time, on target, right? And I every day walking to the Genome Center, I couldn't help but think, wow, we came together to do this, and yet people are dying across the street at MD Anderson, St. Luke's, and so on. And I was committed, upon the completion of the project, to take those skills and technologies that were spawned in large part as a result of the Genome Project, like biochips that can be on this tiny little 1 centimeter square chip that can instantly determine all the genes that are on and off, and sequencing, where you can decipher every letter of a certain piece of DNA, could be used to cure these diseases that these people are dying of. So my biggest commitment, my biggest goal was to take all these technologies, some of which we developed, to study complex diseases like cancer. Cancer is only the 2nd leading cause of death in the States, but it is a devastating way to die. So I was committed to taking my skills to attacking the problem of cancer. And what we do in our lab is essentially what we have done is built a functional genomics platform that brings state-of-the-art genomic tools, bioinformatics. This is a new field where you take computers to figure out what these large data sets mean, to put it together, and then functional studies to identify what are called tumor suppressor microRNAs. And these are microRNAs that are able to suppress the growth of tumors. And through this effort, we have created a unique database that comprehensively describes patterns of microRNA and gene expression in hundreds of cancers, and we have a special focus on cancers in women and children. And we have then a picture that we would never have obtained, without these technologies. So we have a view of the whole genome and how it has changed in the cancer, and we have identified through this work some very exciting microRNAs, a tiny 22-letter RNA that can go into these cells and kill them. And that is hugely exciting.